Yeah, well, we're pulling up the talk. I want to certainly thank Howard and the organizers for inviting me. And for full disclosure, I actually work for a company called Ocell, and that's located in Northern California, but I am a guest researcher at NIH. So my talk is modifying the vaginal microbiome to protect against HIV. And which button? All right, that's not the right button. There we go. So HIV infection around the world, women are becoming one of the most groups that who are infected by HIV. So there's a need for prevention strategies for women. In 2013, almost 60% of all new HIV infections occurred among women, particularly young women and adolescent girls ages 15 to 24. By age 24 in South Africa, 50% of women are infected with HIV versus their male counterparts, where it's only 6%. They're even suggesting to young women, have sex with men your own age because you're less likely to get infected than having sex with older men. So HIV is transmitted across mucosal surfaces. In women, it's generally transmitted across the cervix and vagina. This shows the vagina, and it infects target cells beneath. But as we know, all mucosal surfaces are continuously exposed to a community of microorganisms. Now, we've heard a little bit about mixed communities in the vagina. I'll be talking more about lactobacillus related. So in the vagina and cervix, lactobacillus are found in anywhere from 51% to 90% of women. This shows a beautiful lactobacillus clinging to a vaginal epithelial cell. And the three most common types that are H2O2 producers are Lactobacillus crispatus, Jensenii, and gasseri, with Einers being a non-H2O2 producer. These three strains produce D-lactic, and Einers produces only L-lactic, and D-lactic has been shown to have activity against Gardnerella, which is one of the BV-associated organisms. So the vaginal microbiota is related to health. When you have a low diversity, it can be a defense against urogenital pathogens. You have low vaginal pH. You can produce antimicrobial substances, and you may block pathogens by competitive exclusion. When you have high diversity, and here's your low diversity community, high diversity, you have increased risk of infections, including BV and UTIs, increased preterm birth and other OBGYN complications, and increased inflammation and risk of HIV. So in a recent paper from a group at Harvard that studied South African women, a majority of these women had low abundance of lactobacillus. And this low abundance, together with high diversity, correlated with genital inflammation. So I'm just going to highlight the two types, the Lactobacillus crispatus group. So this is the good lactobacillus in these women and very low diversity. And the last group, which has high levels of Prevotella and Gardnerella, and these women had high diversity. And you can see that the community types separate, so the lactobacillus groups are completely separate from the high diversity. And what was found with these women is the women that had lactobacillus dominated had very low levels of these inflammatory cytokines, whereas women with Prevotella and Gardnerella groups had high levels of the inflammatory cytokines. And again, inflammation is a big risk factor for HIV. So this leads us to Ocell's technology, modifying the vaginal microbiome to prevent HIV infection. So our idea was to engineer one of the good vaginal lactobacilli, and I think the pointer's dying, to make a recombinant live biotherapeutic. Now, this term live biotherapeutic comes from the FDA. This is not our term. It's a term that is given to an organism that now has a health claim. So it's no longer a probiotic, but it's a live biotherapeutic. So we would colonize with this modified lactobacillus. We eventually used a lactobacillus Jensenii H2O2 producer, may have a natural defense, being anti-inflammatory, producing lactic acid, and we genetically modified it to produce an HIV entry inhibitor so we would protect women at the site where infection occurs. So in order to make this live biotherapeutic, we screened a number of different isolates of Crispatus chinsonii and gasseri that were from healthy women. We eventually selected a Lactobacillus chinsonii 1153, and although variations exist in the microbiota over time and in individuals in different geographic settings, you can find Lactobacillus chinsonii, and maybe not this strain, in somewhere between 17 to 41% of women from around the world. We selected this lactobacillus because it had a good growth rate, it produced high levels of D-lactic acid, it adhered well to tissue culture models, and tissue explants, it had activity against Gardnerella and Staphylococcus, and we could transform it. It had no endogenous plasmids, and the Broad and JCVI sequenced the genome for us. So then we wanted to select an inhibitor, and I'm reminded that the audience isn't maybe HIV experts, so when you think about HIV and you're thinking about inhibiting virus entry, you have to remember that HIV is a very diverse virus, and while clade B is the most common in the U.S., clade C is actually the most common worldwide because it's most common in South Africa where most infections occur. So we tested a number of different entry inhibitors, and we eventually selected an inhibitor called cyanovirin N. So cyanovirin N is from a cyanobacterium. It was identified by the National Cancer Institute in a drug screen. It has activity against all subtypes of HIV, and the concentrations that we can produce in lactobacillus are about 272 nanomolar, which is about 3 micrograms of the protein per mil of lactobacillus. And this concentration could inhibit about 85% of HIV from a representative panel of subtypes. And cyanovirin is active at pH, which is the vaginal pH, which can range anywhere from pH 4 when lactobacillus are present to pH 8 when ejaculate is present. So cyanovirin binds to HIV. It has a low affinity and a high affinity binding site. It binds to GP120. It can either bind with a high affinity or the high and low affinity site. And this, just to give you a little perspective, here's a cell, here's HIV, and this is a lactobacillus that's sitting along in the plane. Would be actually much longer than this because it's been cut off in this EM. So we identified a lactobacillus and a strong HIV inhibitor that worked against multiple subtypes. So next we wanted to make a recombinant. So we used a very simple technology. We stably integrated the cyanovirin gene under a ribosomal promoter into the lactobacillus chromosome using homologous recombination. The strain integrates and then resolves, so we end up with a single insertion of the promoter and the gene in lactobacillus, and it produces biologically active protein from this constitutive promoter. So now we wanted to look at whether the strain was fit, and the criteria we used were, first, did our strain produce the same amount of D-lactic acid? So a wild-type strain produces about this much D-lactic acid, and our recombinant strains also produce the same amount. So we hadn't disrupted their basic lactobacillus function of producing lactic acid. We did very basic growth rates to see if our recombinants could grow to the same level as the wild type, and they did. And they were able to produce cyanovirin constitutively throughout their growth cycle. And they also the strains continued to produce hydrogen peroxide. So we'd constructed a fit recombinant. Next, we wanted to test this in an animal model. We initially tested it in mice. Mice aren't really a very good model for lactobacillus, so we moved to primates, and we used a macaque model, and we showed that macaques could be colonized with lactobacillus, although it's to much lower extent than women. So in initial studies, we grew up just a fresh culture. We spun it down, mixed it with hydroxyethyl cellulose, which is used as a placebo, and we applied it to the macaque vaginally. And within 24 hours, we could do a gram stain. This is a Brown and Hobbs modified gram stain, and we could see our lactobacillus in a vaginal smear from the macaques. We then colonized macaques for 5 days with our lactobacillus, and we followed them for 6 weeks. And this is when the animals menstruate. So you can see that the levels of lactobacillus can drop precipitously when the animals are menstruating, sometimes to very low levels. But then they rebound, and eventually these strains will be lost over time without reinoculation. So we showed that our strain could colonize the macaque. Next, we wanted to prove that it was actually making protein. So we colonized macaques, and we did cervical vaginal lavage. So we could see that at day 0, we don't see any of our protein, but as little as 24 hours after colonizing the macaque, we start to see protein being produced from a vaginal lavage, in a vaginal lavage. And at 3 weeks and 6 weeks, this is without any reinoculation, we still continue to see protein being produced. And you can see this one animal has kind of low levels. That animal may have been menstruating during the study. Then we wanted to look at where our protein was localized. So we took biopsies from macaques and performed immunohistochemistry for the cyanovirin protein. And this is, again, vaginal surface and the vaginal lumen. And you can see the brown stain. If you look very closely, it's quite associated with the bacteria on the lumen and vaginal surface. So we showed that our lactobacillus could colonize a macaque, could make recombinant protein, and we showed where that protein was, and we believe that protein was in the right place to prevent HIV infection. So now we wanted to challenge with a simian HIV to test whether the infection could be prevented. So we did a 2-arm study. We took 12 macaques that received our lactobacillus-expressing cyanovirin. Again, this was a live culture. And we had 12 control animals. 8 of these actually received the HEC placebo, and we had 4 that received no gel at all. Macaques were challenged each week for 6 weeks, and they were monitored for viral infection. These are called repeated dose challenge models, and they measure the rate of infection per exposure. The infection rate that we started with was about 33%, so 1 in 3 animals should get infected at every exposure, and this is about 1,000 times higher than the HIV infection seen in humans, and then we monitor time to infection. So this is what happened. If you just look after the first 3 weeks, you can see that our experimental group, which got lactobacillus, is being infected at a much slower rate. And when we ended the study, it took us 31 challenges to get 11 animals infected. So that's a 35% infection rate. In this experiment, it took us 61 challenges to get 8 animals infected. That's a 13% infection rate. In this case, 4 animals remained uninfected and 1 remained uninfected here. Oops. So if you look at this on a Kaplan-Meier plot, you can see that there's nice separation. And our p-value, so we had a 63% reduction in acquisition, and our p-value was 0.004. We also saw a 6-fold reduction in viral load in animals with breakthrough infection. So this was unexpected. We think this may be related to the fact that the lactobacillus may be anti-inflammatory, so the macaques may have had a less initial nidus of infection, but we don't have any evidence to prove that yet. So we had efficacy in our challenge model. Now we wanted to move forward with a formulation that could be used in the real world because we're not going to apply live bacteria to it wouldn't be very practical. So we formulated our lactobacillus as a prototype vaginal disintegrating tablet. So the tablets are very potent, about 10 to the 11th organisms per gram. They're actually about 100 milligrams, so 5 times 10 to the 10th per tablet. And we showed that we could colonize macaques. If you look like at 3 days, they're really, really high levels, about the same as the tablet, but we wanted to be sure we were measuring actual colonization, so we look at 3 weeks. And you can see quite a range. It's very similar to the levels that we saw when applying live bacteria. This animal was menstruating on the day of sampling. We had 1 animal that was only detectable by PCR. So we repeated our challenge study with a smaller group of animals applying the vaginal tablets. And in this case, we had 4 animals readily colonized in our lactobacillus group and 3 animals in the placebo group. And after one challenge, and we had one animal that did not colonize until like the second or third week. So all the placebo animals were infected on the first challenge, and the one animal that was not colonized was infected. So although we didn't reach statistical significance, we have the same trend as our previous study. And a little snapshot of the microbiota, if we look at this animal that was well colonized with lactobacillus and we just look at the bacilli group, we can see that most of the bacilli in the animal that was colonized are lactobacillus, and we actually confirmed that this was our strain by sequencing the inserted cassette. And the placebo animals, you can see, have very small levels of lactobacillus, shown here in purple, and they have a lot of enterococci and others in their bacilli component. So to summarize, we identified a need, which was to develop HIV prevention for women. We selected a lactobacillus, which is a vaginal colonizer. We selected an HIV entry inhibitor. We constructed a recombinant. We tested its fitness. We tested colonization in a macaque model and protein expression and efficacy. We formulated this into a product, and I didn't say this, but our product is stable for about a year at 4 degrees and 25 degrees. We're working to improve high temperature stability, and we tested efficacy of the tablet. So we're now moving this into a IND pathway. We're preparing a pre-IND for the FDA. We're finishing the formulation process and methods development. We're transferring to an industrial partner, which we've identified for manufacturing the product. And we're doing preclinical safety testing in the vagina. This usually includes a rabbit vaginal irritation model, and we'll be speaking to the FDA about what other models may be appropriate to test safety of this product. Since not all mammals are colonized with lactobacillus, some models may be more or less appropriate to test for safety. And then we'll prepare an IND package for the FDA. So far, we've had a good safety profile. We've done some mice studies, and we're just starting rabbit studies. The product was not immunogenic in the macaques, and also we showed that there was no evidence of inflammatory cytokines in the macaques, no evidence of toxicity. We could potentially improve vaginal health. We might improve adherence in the microbicide field or of HIV prevention. Women have a hard time using products that are coitally dependent, where they have to use it every time they have sex. And a product like this could be used female-controlled and independent of coitus. And economically, these will be very inexpensive to produce and hopefully will improve long-term stability so that a woman could get a supply of the product and use it. Obviously, one of the big factors that has been talked about is bacterial vaginosis and how to best colonize women who start out with a BV condition. We're doing a study with non-recombinant lactobacillus in women with BV. So we're hoping to learn a lot about factors that improve colonization. And one thing we found already is if you can clear that BV biofilm to begin with, you can colonize a woman pretty well with lactobacillus. If you don't clear the biofilm, it just comes right back, and it's harder for lactobacillus to get in there. So effective, whether it's antibiotic or combination antibiotic treatment, is really important to improve infection. That's it. I want to thank members of our former members and our funders.
Thank you. Very nice talk. What do you expect for the possibility of having an HIV strain selected that doesn't bind to this protein?
So cyanovirin binds actually to glycosylation sites. So it's very unusual for, you can make a strain in the lab that becomes resistant over time to cyanovirin, but because it's going to be exposed to the virus for very short periods of time, it shouldn't become resistant. And again, we're always on the lookout for new and better proteins, even some small antibodies that may be very potent.
Cyanovirin was actually discovered here on this campus and worked up by Barry O'Keefe, and it's non-immunogenic. They've not seen any immune response to it, and it's active against many different viruses. So are there any other virus models that you think that this is going to be tested in with your lactobacillus?
I know cyanovirin has activity against a lot of different viruses, but we haven't thought of any that work with the vaginal space and lactobacillus anyway.